Welcome to The Explainer. Today, we are going to zero in on a single, really fascinating piece of our genetic code. It's a variation that has some pretty profound implications for how our brains process reward, and, that in turn shapes how we behave. We're talking about something called the Taq1A polymorphism. So what's our game plan for unpacking this? Well, we're going to start with the basics. What even is this polymorphism? Then we'll dive into its genetic roots before mapping out how it can actually shape our behavior. After that, we'll explore the main scientific theories that try to explain these connections, look at the real-world clinical side of things, and then we'll wrap up with a critical look at what the science can and can't tell us.
All right, first up, let's meet our central player, the Taq1A polymorphism, and its two really important versions, which we call alleles. Okay, so this specific genetic marker, its official name is RS1800497, is what scientists call a single nucleotide polymorphism, or just SNP for short. Now, I know that sounds complicated, but you can really just think of it as a common one-letter typo in our DNA. And this tiny little variation is a huge deal because it has a major impact on our dopamine system, which is basically the brain's master circuit for all things motivation and reward. And this right here is where we see the fundamental split. You see, depending on whether you carry the A1 or the A2 version of this gene, your brain's reward machinery can be wired pretty differently. The A1 allele, that one's linked to having fewer dopamine receptors, which creates what scientists have dubbed a reward deficiency state. On the other hand, the A2 allele is associated with the more typical brain setup. So we know there are these two versions of the gene, but to really get how they work, we need to zoom way in, down to the molecular level, and see exactly where this variation is and how it's able to exert its influence. And here's a really crucial scientific update. For years, everybody thought this SNP was located inside the dopamine D2 receptor gene, the DRD2 gene. But we now know it's actually in a neighboring gene called ANKK1. However, and this is the key part, it's so close to DRD2 that they're almost always inherited together, like a package deal. So even though it's technically an ANKK1, its effects are overwhelmingly felt on that D2 receptor system. Now, since we all get one copy of a gene from each parent, there are three possible combos, or genotypes you can have. You can end up with two copies of A1, one of each, or two copies of A2. And for most of the research we're going to talk about, anyone with at least one copy of the A1 allele is considered an A1 carrier, because it turns out even just one copy is enough to have a measurable effect. Okay, so let's connect that genetic code to stuff we can actually see and measure, what scientists call the phenotype. So this is the number that really matters. In people who carry that A1 allele, brain imaging studies consistently show a pretty significant drop, about 20 to 40%, in the number of available D2 dopamine receptors. And this is happening in a key reward area of the brain called the striatum. Think of it as a major hub for motivation and habits. This isn't some subtle thing. It's a major, measurable biological difference. And as you can see in this table, that one biological difference creates this whole cascade of consequences. If you look down the column for A1 allele carriers, that reduction in D2 receptors leads to a blunted, kind of muted sense of reward at baseline. This can mean they have lower sensitivity to natural rewards, but a higher drive for artificial ones, like drugs. And this maps directly onto things like increased impulsivity, less efficient executive control, and a demonstrably higher risk for addiction. So we've seen the what, that link between the gene and behavior. But how does this actually happen? Let's dig into the three main scientific hypotheses that try to explain these connections. The leading theory is called the reward deficiency hypothesis. The idea is that having fewer D2 receptors creates this chronically low baseline level of dopamine. To compensate for that, the brain basically pushes the person to seek out really intense experiences. Thrilling activities, super flavorful foods, substance use, just to get that reward signal up to what feels normal. It's almost like their brain is always looking for a fix just to overcome a natural deficit. The second model is all about salience, which is just a fancy word for what our brain flags as important. D2 receptors are supposed to help put the brakes on things, to control what we pay attention to. With fewer of them, the brain of an A1 carrier might struggle with that control, causing it to assign this exaggerated importance or incentive salience to things like drug cues or other high-intensity rewards.
And the third hypothesis zooms in on the brain's circuitry. It suggests that having less D2 signaling weakens that critical communication loop between our prefrontal cortex, which is like the brain's CEO, and the striatum, our habit center. When that connection is weak, it can lead to poor impulse control and a tendency to fall back on automatic, habitual behaviors instead of more thoughtful, goal-oriented ones. So, we have these three powerful theories for how the A1 allele works. Now let's bring it back to the real world and see how these biological mechanisms show up as actual clinical conditions. The evidence is definitely strongest when it comes to addiction. There are really consistent links to a whole range of substance use disorders. But the implications don't just stop there. That impulsivity and weakened executive control we've been talking about, those are hallmark traits of conditions like ADHD. And that reward deficiency model provides a really compelling way to understand certain patterns of obesity, where you might see overeating as a way to chase that reward signal from food.
This slide does a nice job of pulling it all together, showing how these different scientific models each give us a unique angle on that A1 phenotype. And they aren't mutually exclusive. In fact, they probably all work together. You could have a baseline reward deficiency, combined with an executive system that struggles to control impulses, especially when it's faced with cues that your brain has already flagged as being extremely important.
Okay, finally, it's so important that we step back and look at all this research with a critical eye. What do we know for sure? What are the limits? And where is this whole field going? The strengths here are pretty clear. That biological finding of fewer D2 receptors is very, very solid, and the link to addiction has been shown again and again. But we have to be cautious. This is just one gene out of many that influence these complex behaviors. Its effect is modest, and environmental factors play a huge role. Having the A1 allele is a risk factor. It is absolutely not a deterministic life sentence.
To put a number on it, scientists would probably assign a high confidence score, something like a 0.8 out of 1, to that core biological link between the A1 allele and having fewer D2 receptors. That connection is considered really well established.
However, the confidence in using this one gene all by itself to predict a complex outcome like addiction, well, that's more moderate, maybe around a 0.6. It gives us useful information for sure, but it's only one piece of a much bigger, more complicated puzzle.
The future of this research is really all about integration. Instead of looking at Taq1A all by itself, scientists are building what they call polygenic risk scores. These scores combine the effects of many different genes to create a much more accurate picture of someone's genetic risk. And the ultimate goal here is personalized medicine, where you could use this genetic info, along with brain scans and behavioral tests, to tailor therapies specifically for an individual.
So if you take one thing away from our explainer today, it should be this really clear chain of events. The A1 allele is strongly linked to having fewer dopamine D2 receptors. This helps create a state of reward deficiency, and that in turn increases an individual's risk for impulsivity and addictive behaviors. And this leaves us with a final and very important question to think about. As we get better and better at figuring out the genetic roots of our behavior, we really have to ask, what are the ethical guidelines for using this kind of predictive knowledge, both in the clinic and just in society in general? Definitely something to chew on. Thanks for tuning in.